All right, welcome back. So we are at our last video here of the chapter 13. I know there's only 4 sections, so there might be some intermittent little help tutorial videos or things like that, but there's really only 4 sections, 4 main lectures. And we're at the last one here. It's going to be about gene regulation and gene expression. So we've done a lot of talk about what genes are and how they're basically expressed through making some mRNA and letting it get translated by the ribosome and the cytoplasm and so on, and how they make proteins. But we didn't really talk about like what controls all that. When does it happen? Like why does it happen sometimes, and how often does it happen, and such like that. Like who's got their hand on the throttle, if you will, about that. And so that's an interesting topic of study right now. And so this is a great way to finish off our topic here, is about regulation. And thinking of that, we oftentimes have to regulate our own behavior, and we have to regulate our own lives and be in control. So my worship thought is kind of centered around that. You know, one of the fruits of the Spirit is self-control, and we're called to control ourselves. Yeah, I think that's a blessing that God gave us, that freedom of choice and that privilege to control ourselves. And oftentimes I know it's difficult, but there is a system in place that God has. In Philippians 4:8, one verse that I kind of like that says, finally, brothers and sisters, whatever is true, whatever is noble, whatever is right, whatever is pure, whatever is lovely, whatever is admirable, if anything is excellent or praiseworthy, think about such things. And I think what that does is it kind of sets off a chain reaction of thoughts that are also, or that will continue the thoughts of nobility, rightness, purity, love, and things that are admirable and excellent and praiseworthy. You know, you can even see it nowadays in the world with the coronavirus. You know, a lot of people are really shunning the negative talk, whether it's Facebook or any kind of social media websites. I have a Nextdoor app I usually visit that talks about things that are happening in the neighborhood, and everyone's shutting negative comments down because they don't want to hear it, you know, because all it does is just fuel more negativity. And same with, you know, news. You watch the news, and that's kind of what you get, right? You get one side fighting the other, and all it does is just kind of fuel the next negative thought. But positive thoughts will have the same impact. And so if we're looking for ways to have healthy thoughts and live a healthy lifestyle, then at least emotional health, then that's what the Bible clearly gives you the recipe. And so I know I'm not perfect either, and so maybe there's some things, shows on Netflix or things like that, or news, like I said, just negative news stuff, comment sections that I read that I shouldn't read them, you know, because all it does is entertain that negative side that sometimes can be entertaining for us, and that may be the road we want to go down, complaining and things like that, but it gets us nowhere. You know, when we put that stuff aside and we consider that only which is right and noble and pure, then we don't have to ask questions, well, is this right or is this wrong, or don't worry about it, because if it's not right and if it's not lovely, it doesn't mean you're not going to heaven, because, you know, I'm not talking about a heaven or hell thing, a salvation thing. That's been accomplished at the cross. What I'm talking about is how we respond to our choices and the self-control that we have. But that being said, let's talk about how cells do that, how they control themselves. And so let's switch to another camera angle here. We're on 13.4, gene regulation and expression. Remember, all these genes code for traits like hair color, eye color, height, and just the famous examples. But everything about you is essentially a trait. Some of it's visible, some of it's not. But all your genes are going to code for proteins. And but again, how do those genes get regulated? You know, we have thousands of genes on our chromosomes, and how do they get, you know, the individual genes, how do they get turned on or turned off or whatever? And so this is our, let me go back to my other camera here. You want a little snapshot of our, something happened. There we go. Maybe that's it. Okay, so 13.4, we basically have gene regulation and expression. We're just going to look at pretty much how that's done in a prokaryotic cell and then a eukaryotic cell, since those are basically your two types of cells. Remember, we are eukaryotic organisms, but we're even more special than the fact that we're just eukaryotic. We are multicellular organisms, so we have a lot going on. Our brain cells, or they're nerve cells, if you will, need to do different stuff than our muscle cells, which need to do different stuff than our skin cells. And so, but yet they all have the same DNA. They all have 23 chapters of our DNA from our mom and our dad, our mom's version and our dad's version. And so how do those cells regulate so that they're turning the right genes on and the right genes off? And so in other words, what's the control mechanisms for multicellular organisms that have to have differentiation occur in their cells so that you can grow different limbs and have different organs that do different jobs? And so let's get started. Just like the last lecture, I'm going to use my PowerPoint here. And so I'm just going to go ahead and share my desktop with you here. Right, and minimize my software, minimize that, go back up to our PowerPoint, and here we go. Again, basically we're looking at how are prokaryotic genes regulated, how are genes regulated in the eukaryotic cell, and then what really controls the development of the cells and tissues in large multicellular organisms. And so if you read this, think about it, basically they're saying, hey, like if you think about a library, a bunch of how-to books, would you ever need to know all those books at the same time? Would you be able to need to access those all at the same time? Of course not. If you want to fix a leaky faucet, you're going to go to the plumbing section and read that book. If you're going to fix do some landscaping stuff, you're going to go to the landscaping section and grab the landscaping book. Now picture a little tiny bacterium like E. coli, which is super small. It's going to have like 4,000 genes. Most of its genes code for proteins that do everything from building cell walls to breaking down food. Do you think E. coli uses literally all 4,000 of those genes at the same time? The answer is, of course, no. It's a rhetorical question. And so how are prokaryotic genes regulated? Because that's what this is. E. coli is a bacterium that is prokaryotic. If you don't remember what that is, prokarry, no carry. Prokarry, no carry, no nucleus, and it's inside of it for its DNA. So the DNA is just floating around in the cell, in the cytoplasm, and here it is all spilled out. Isn't this a really cool electron micrograph of an E. coli showing all of the DNA spilled out? I think that's pretty cool. So anyways, I'm just going to jump right to the sentence here, the key question, so this video can be as short as possible. You can do all this reading yourself, and in fact I encourage you to do that. Some of it I'll read word for word, but most of this reading you should do yourself. I'm rephrasing it myself so that you have a different source, a place to hear it from. But this is a key sentence. It answers this key question, so let's read it word for word. DNA binding proteins in prokaryotes regulate genes by controlling transcription. Oh, that's a power-packed sentence right there, but a lot is packed into that with such few words, so we really need to unpack that really. DNA, let's break this down. DNA binding proteins. Now this is just interesting right here, okay? What does DNA do? Well, so far we've learned that DNA literally has the code for the ribosome or the cell to make proteins. That's like essentially all DNA is for. It literally just has the information, the right sequences, so that the cell knows what proteins to make. That's what this whole chapter is about, is making proteins. But yet, to control genes, turning on or turning off, DNA has these binding proteins that attach to the DNA. So literally the stuff that DNA codes for attaches to the DNA to help control some of that. And so they're called DNA binding proteins, and they literally bind chemically to the DNA, and they're going to regulate. They're going to regulate the genes by controlling transcription. All right, you already know what transcription is, right? That's the making of the RNA from the DNA. And so these binding proteins are going to influence that transcription process. Okay, so how does that work? One of the keys to gene transcription is the organization of genes into what are called operons. An operon is essentially a group of genes that are regulated together. The genes in an operon usually have related functions. And for E. coli specifically here, there's a cluster of three genes actually that together must be turned on before the bacterium can do certain process which is to break down lactose as food. I know we did a whole 2 chapters, one on photosynthesis of how plants make glucose, and one on how humans and other organisms break down glucose for energy in the cellular respiration chapter. And so we did a lot of talk about glucose, but lactose is another sugar that organisms can digest to get energy from, and it has a chemical structure sort of similar to glucose, just some minor differences. We're not going to go into that, but the point is lactose is a sugar just like glucose in the sense that it provides energy. Even for us as babies, when they're breastfeeding, they have enzymes that will help break down the lactose so that the lactose can be quickly converted to energy. As adults, when we're weaned and we're no longer breastfeeding, we don't carry those enzymes, and so drinking milk actually does not necessarily provide us with the lactose in a beneficial way, and some people can have negative side effects because of that. Some people are lactose intolerant, and in fact, some would argue that we all are after we're weaned on milk. But anyways, enough of that tangent. The point is, for E. coli, in order for them to digest lactose, there actually has to be those 3 genes in that operon, those cluster of 3 genes that make up that operon. Those have to be turned on together, all together, in order for the organism to do that. And so here's a nice illustration of how that happens. And it's actually called the lac operon. Lac stands for lactose. So the lactose operon, or the lac operon, works like this. We're going to be referring to this image here and this image here, as well as this image. So we got 3 images we're going to refer to here. And so basically, like I said, in a nutshell, lactose is a sugar that's actually made up of glucose and galactose together. So glucose and galactose come together to make a sugar called lactose. And to use it for food, the bacteria has to transport it across its cell membrane, break those bonds so that between the glucose and galactose, so that it can digest them individually. And so the tasks are performed by proteins, literally coded for by the genes in that lac operon. So these lac genes here are responsible for making the proteins that are going to do all of that work of getting the lactose in the cell, breaking the bonds between glucose and galactose so that they can be digested individually for energy, and all of that together. In other words, just know that lactose can't be digested unless these lac genes are translated into their proteins. Remember, it's actually not the DNA. This is DNA. This dark blue line here, this whole section of stuff here, this is DNA. I know we should technically see 2 strands, but we're quite a way zoomed out here. And DNA doesn't actually get translated. It's the RNA that gets translated, right? But so this is just the best word I could choose for the moment. But so how does this work then? Let's continue to look at this. We basically have these lac genes here. We have something called a lac repressor gene. Repressor means to slow down. Repressor means to slow down or stop. Pretty much in this case means stop. And so there's actually a gene here. This orange section of DNA is a gene that codes for a protein, and it's right here. It codes for this repressor protein that is actually going to attach to this site right here called the operator. This is where the repressor is going to bind. Again, this is a repressor protein, so we call it the repressor. All right, it's going to stop this process. It's going to attach here, and as long as that repressor protein is here, which is what you see here, as long as it's attached to the operator, as long as this repressor protein is attached to that operator like you see it here, then RNA polymerase, which you remember makes mRNA, can't read this. It can't read the DNA and make an mRNA strand. It's not going to make a transcript. It's not. So if you don't even start the process of transcription, translation won't even occur, right? Because you have nothing to read. So again, this is an awesome mechanism in a way for, this is just the start of the story, by the way. Stay with me here. This is an awesome way to stop the process of these lac genes being interpreted or read. Let's just put it that way, okay? If these lac genes cannot be transcribed into mRNA, then you're not going to get the proteins that those genes code for. So RNA polymerase will just run into this repressor protein and it'll just give up. It loses the battle. All right. Now you also have this RNA polymerase promoter. This is the promoter section. This is where RNA polymerase binds. And so even though that's there, that will allow the RNA polymerase to attach. Okay, the promoter's there. That's a section of DNA. Again, a section of DNA or a gene, a whole section of DNA that does something. We call it a gene. It's a gene that allows RNA polymerase to attach. So it'll do that, but again, it's not going to keep moving forward and reading these lac genes. It can't do that if that oppressor is there or repressor is there. And so check this out though. Here's the cool part of the final of the story here. If lactose is present, if these are these little green things here, if lactose sugar is present, what it'll do is it'll attach to the repressor protein. Okay? It'll bind to the repressor protein, this chemical reaction. So it'll bind to the repressor protein, changing its shape. And if it changes its shape, this repressor protein will not bind to the operator. Does that make sense? It's almost like a lock and a key. If you put these lactose molecules on this repressor protein, it kind of changes the shape of the key and it won't fit inside of this lock. That's an analogy we'll use later on too. But that's kind of how enzymes work, and this isn't necessarily like an enzyme type substrate type system, but kind of the same idea. You change the shape of this protein here, this repressor protein, and it's not going to attach anymore to the operator. And then RNA polymerase is going to do its job. It's going to run through the DNA and read it. And as it's reading it, remember, it's going to be making an mRNA strand. Now that mRNA strand obviously will get taken into the cytoplasm and be translated into a protein. And so you'll have your protein. So as long as lactose is present, then the lac genes will be interpreted. They can be read and they can make the, guess what this mRNA strand codes for again? It codes for, remember back at the beginning, it codes for the proteins that are going to digest lactose. So what a cool system, huh? That when lactose is present, these genes are turned on essentially, so that you have the enzymes to break down the lactose. But if lactose is not present, then there's no reason to have these lac genes read. What's the point? Right? You don't need the proteins to break down lactose if you don't have any lactose to begin with. So this is a really cool kind of feedback system that is just kind of naturally built in to the cell. It's very basic. This is how prokaryotes do their job. All right. So again, we said we were gonna look at how prokaryotes do gene regulation, and then how eukaryotes are going to do gene regulation. So we're about to look at eukaryotes and how they do that work. And then we'll talk about how multicellular organisms are regulated as well. So back to our slide here. Eukaryotic gene regulation is very similar. In fact, what they say here is that the general principles of gene regulation in prokaryotes also apply to eukaryotes, although there are some differences. And so let's go over those. Most of the eukaryotic genes are controlled individually and have more like complex regulatory sequences than those of the lac repressor system. The lac repressor system was basically just you had the operator, which can be plugged by the repressor protein or not. So pretty simple system, but in eukaryotic cells, they're a little more difficult. You got these multiple transcription factors, which are also proteins, by the way. It's kind of interesting. Remember, this DNA is designed to make proteins, and here you go. You have some proteins attaching to the DNA. And what they do is they attach in a certain way to sections of the DNA called enhancers. It's kind of like the promoter. It's, the promoter is what allows the RNA polymerase to attach and get started. It's kind of like the place where it all begins, because DNA, remember, is full of thousands of genes. And just like a book can be full of thousands of chapters, how do you know when a chapter ends or begins, right? Like a table of contents. And that's kind of what this is. This enhancer is like saying, hey, this is the beginning of the chapter. Let's start transcribing. And so RNA polymerase knows to start actually by binding to this area called the TATA box, but this enhancer is a region of DNA that allows the transcription factors to bind. And when they do that, and they also bind to the TATA box, and all of this changes shape and fits, and everything is nice, and the chemical reactions take place so that the RNA polymerase can finally do its job. And so I know I said that quick, so let's back up again and talk about these things individually. The figure here shows several features of a typical eukaryotic gene. So we would call this whole thing like a eukaryotic gene. And one of the most interesting is this TATA box. We call it a TATA box. It kind of has a funny name. You'll probably never forget that. A lot of students pick a few things they remember from biology, and the TATA box is just kind of funny sounding, so they remember that. And it's basically a short region of DNA. Not going to call it a gene. It's just a short region of DNA, about literally 25, maybe 30 base pairs long. All right, so not that big. And that short region is going to be the start. It's going to be the start of a gene. It's going to be like the opening of the chapter, the intro, basically. And it's going to contain the sequence TATATA or TATAAA. And so you pronounce both those TATA, right, if you were to actually try to pronounce them. So that's why it's called the TATA box. And the TATA box is going to bind to that protein there. It's going to bind to that protein that helps position the RNA polymerase by marking a point just before the beginning of a gene. Otherwise, RNA polymerase is kind of walking around like, hey, where do I start? Where do I start? Well, when he sees this guy here bonded to the TATA box, he knows that that's where I start. That's where he should start. So gene expression, remember, it can be regulated at a number of levels, like, the transcription process could be regulated at the translation process. But in this case, it's being regulated at the transcription process, right? Like, it's being regulated, whether this gene is read or not, is regulated by the transcription process, because that's what RNA polymerase does, is it transcribes messenger RNA, remember? It transcribes messenger RNA. And then that messenger RNA still has to be translated. So that's like another step in the system that you could technically control or regulate whether or not you have a protein at the end of the day. But this one is controlling the transcription. So that's why these are called transcription factors. And so again, how are genes regulated in eukaryotic cells? What's the main way? Well, it's by binding DNA sequences in the regulatory regions of eukaryotic genes. Transcription factors, that's these proteins here, control the expression of those genes. Okay? By binding DNA sequences in the regulatory regions of eukaryotic genes, transcription factors will control the expression of those genes. So it's all about these transcription factors controlling the expression of these genes by whether or not they're binding to these regions here, the TATA box and the enhancer. So the enhancer can be large, it can be small, depends on what gene you're talking about and what organism and all that. But in general, you just have these general terms that we say here. The enhancer, these are transcription factors. And again, these may be different molecules depending on what gene or organism. But we always have this TATA box as well. And then, so there you go. Again, once all of this comes together, it literally comes down to two terms you hear a lot in biology and A&P, structure and function. And when this has the right structure, when it has the right shape, literally, as these molecules bind and bond to each other chemically, then they create this special shape, which is kind of like the green light or the signal for the RNA polymerase to run and do its job, which, remember again, is transcription. RNA polymerase does the job of transcribing RNA, messenger RNA. The direction of transcription will be this way, and then it'll eventually, remember, run into a stop codon and all that. Well, that was chapter 13.2. So, okay. So just a couple things to highlight here. So let's see. Promoters, they're going to have multiple binding sites for transcription factors, each of which can influence transcription. So certain factors, these purple transcription factors, certain of them can literally turn on or activate like scores of genes at once, right? So it's like if you have those transcription factors, all of a sudden all these genes get transcribed, okay? Like it's pretty dramatic. Literally dramatically changing patterns of gene expression in the cell. Other factors may, for example, respond to chemical signals. Like, for example, steroid hormones, they're chemical messengers, literally, that enter cells, and then they bind to receptor proteins. And then that complex of the hormone and the receptor protein all attached to each other, those big complexes then act as transcription factors. So like one of these, in this case we're talking about right now, one of these purple things would be a protein and a steroid together, bonded together. Well, when the steroid is bonded to the protein, then it'll act as a transcription factor, touching the enhancer, getting, again, all this process started. So having the hormone would literally turn the gene on or turn the gene off, all right? And so eukaryotic gene expression can be regulated by those factors. Other factors too, like the exit of an mRNA molecule from the nucleus, the stability of mRNA, or even the breakdown of a gene's protein products. So anyway, the point is there is a system in place for eukaryotes to turn genes on and turn genes off. They don't just get translated and turned into proteins randomly and just arbitrarily. There's a reason and rhyme and reason for genes to be turned on and turned off. Otherwise, it'd be crazy if cells were just constantly doing whatever the DNA said to do. Because you have to remember, let me come to get away from the book for a second. You have to remember that every cell in your body has all of your DNA to do anything that any cell in your body does, which is kind of weird to think about, that one of my skin cells has the ability to do things that my brain cell can do, or have the ability to do things that my muscle cells can do. It's just a matter of those genes aren't turned on or turned off in that case. And so what made my cells go one way or go another way and decide to be a nerve? Because at one time I was just one cell. And then I became 2, and then I became 4, and then I became 8, and then I became 16, and all my cells kept replicating and duplicating. But why aren't all of my millions of cells the same? Because they've specialized. If you guys remember from chapter, I think it was 11 or 10, we talked about cell mitosis. We talked about how when you start out as an embryo, you've got these different cells, like totipotent cells can be anything. Pluripotent cells can be basically any type of tissue that they are. And then multipotent cells can be basically just themselves, basically. I think I said that right. Point is, you've got these stem cells that can differentiate. And so that leads us into our next conversation here, which is cell specialization. And this is another interesting little process, but fear not, if you're a visual learner like me, we have a figure to study. I love visual aids like this. And so, and this right here is this paragraph. All it is just repeating what I just said, but they do bring a better example. So here's a great example. Think for a moment. The genes that code for liver enzymes, for example, are not expressed in nerve cells, right? Keratin, which is an important protein in your skin and your hair and nails, keratin is not produced in your blood cells. So blood cells don't read the DNA to make keratin. That would be pointless. They don't need it. So cell specialization requires genetic specialization, right? Cell specialization requires genetic specialization, if you will. But yet again, all the cells carry the same genetic code. So how do you pick and choose which chapters of the DNA you're going to read? And so here's one way. Here's a mechanism called RNA interference. RNA interference. This is some good stuff to read word for word. For years, biologists wondered why cells contain lots of little small RNA molecules, literally only a few dozen base pairs long, so like 24 base pairs long. You just have this section of RNA. If you're wondering why it's looks like it's DNA because it's two-stranded, let me keep reading this sentence. In the last decade of as figure 13.18 shows, after they are produced by transcription, the small interfering RNA molecules fold into double-stranded hairpin loops. So this is essentially just a small section of a couple dozen or a few dozen base pairs long of some RNA. And after it gets transcribed, again, what they've noticed is that it wasn't a particular important RNA. Like they noticed it didn't contribute. It wasn't like messenger RNA. It didn't act like any kind of tRNA bringing amino acids. It didn't act like ribosomal RNA, which makes up the ribosome. So of the three main types of RNA, this really didn't have a purpose. It didn't really fit any of the categories. And so scientists were really studying that. And then in the last decade, they made some important discoveries about those small RNA molecules. And so here's again what they noticed. They noticed that after they're produced by transcription, those small we call them interfering RNA molecules, they folded into these double-stranded hairpin loops. And then an enzyme called the dicer enzyme cuts or dices these double-stranded loops into what are called microRNA. MicroRNA. You abbreviate that with a lowercase m and a lowercase I, almost like miles, and then all caps RNA. So that's microRNA. And that's what you would get after the dicer did its job. So the dicer enzyme would come along here and it would dice or cut these little sections of microRNAs. So you have these little bitty sections of double-stranded microRNA. Now check this out. This is where it gets really interesting. The two strands of the loops then separated, and one of the microRNA pieces attached to a cluster of proteins to form what is known as a silencing complex. So one of these strands would break off from the other and come together with these proteins here, a cluster of proteins, to form this huge silencing complex. And just like the name implies, it literally silences this section of our mRNA, rendering this whole entire mRNA strand useless. And so guess what? After it renders this section of RNA strand useless, as we studied in the last chapter, when you mess up the sequence in any way, even though this sequence is all intact and this sequence is all intact, you're not going to get a protein because all of this stuff has to be fluid. You can't have any mistakes. You can't just silence a whole region of messenger RNA because the whole messenger RNA needs to get read. So this would severely disrupt that process, making you not get a protein. All right. And so this is one way that the cell has the ability to stop mRNA from being translated. And so that would be at the translation process. Okay, so let's say the mRNA could get transcribed, but you still wanted to stop it from making a protein. Well, here's your last chance to do that is by using these microRNA, these pieces of microRNA that will come together with proteins that make a silencing complex. All right, at first, RNA interface seemed to be a rare event found only in a few plants and other species. It's actually now clear that RNA interface is found throughout the living world and that it even plays a role in human growth and development. So some pretty neat stuff there. One thing, I gave you an assignment here, just these 3 little simple questions. Basically read this paragraph to kind of explain what this table is telling you. I really like you guys to get the exposure of how to analyze some data in a graph and then be able to answer questions related to the data table. So make sure you read like the header up here, the header for the different columns, and then look at the rows here. And if you read this in pretty good depth, without just jumping straight to the questions, trust me, then you should be able to answer the questions. But don't just try to answer these questions right away. Like, read this paragraph and then look at the data table and then try to read the questions, because you'll get better answers. The discovery of that RNA interference technology has made it possible for, again, researchers to switch genes on and off at will, which was related to my kind of worship thought I was telling you guys about, how, our minds will go certain ways, right? Whether we activate certain thoughts or deactivate certain thoughts. And so that's almost influenced by things that we hear, think, and say, right? By input. And so basically by certain inputs, we can turn on or off certain genes, which is kind of interesting. And so it holds the promises of allowing scientists to turn off the expression of genes from, check this out, viruses. So there you go. I don't know if you've watched my coronavirus video that I gave you guys just for fun, to show you how really simple the virus really is and what it does, just kind of give you guys a little empowerment. You can see that here you go. Here's one of your mechanisms for stopping the virus. Literally, once it, in fact, I think I still have that picture. So hopefully you watch the video so this picture doesn't look super scary. But once the coronavirus attaches to these receptors, which again, remember, allows the coronavirus to enter the cell, then inside the lung, was one of the cells in your lung, then the coronavirus will spill its RNA, right? All this RNA here, it'll spill it out. And then of course it's going to get, copied a bunch of times, and then it's going to get translated into these proteins, which were literally the proteins on the outside of the coronavirus, the ones that help it attach to the receptor site, which allow it in. So if you can stop this translation process to these proteins, it doesn't matter that the coronavirus attaches here and it gets into your cell, and who cares? You got the coronavirus in your cell, and even the fact that your RNA-dependent RNA polymerase will make a bunch of these RNA strands, who cares? You've got a mechanism that is going to stop the translation process. So you'll have all these RNAs, but they're not going to get translated, because you've got something to silence some section of RNA so that you don't get these proteins, right? Then you're going to have all this not really be efficient. And so that's just a theory. I am not an epidemiologist or a medical professional, so please don't quote me on that. I'm just these are ideas that obviously could be brought to fruition, and you guys, I'm sure, could make the same inferences, right? Also, speaking on that real quick, I know, sorry, we're on a tangent here, but back to the mutation lesson. One thing that is done with the current treatment, I found out as I looked more into it, was this drug called remdesivir, which literally replaces some of the nucleotides with not different nucleotides. So it's sort of like a mutation, but it adds a chemical, it adds a compound that looks really similar to a nucleotide, but it's not. It's not adenine, guanine, uracil, thymine, or whatever. It's none of those nucleotides, but it's a compound that looks really, really similar. And it's enough that will make the DNA not be able to be transcribed into any kind of dangerous RNA. And so that's something that they know works. Okay, so here we are talking about genetic control of multicellular organisms. So how do we get this differentiation? So you start out with embryo. This is literally what we start out looking like. Wait, hold on. Let me make sure. Nope, sorry. This is a mouse. This is a scanning electron micrograph showing a mouse embryo undergoing cell differentiation about 23 days after conception. But what's funny, what I wanted to say is, this looks a lot like us. I'm kind of laughing and joking and kind of meant to say that, and I'm glad I said that it kind of looks like us, because literally at this stage, a mouse, a human being, you pick it. There's a large number of organisms that would literally kind of look like this, having some kind of headish thing, some kind of armish thing, a leggish thing, and a tailish thing, right? That's kind of what organisms tend to look like in their early stages, and that's because of these genes called homeotic genes. Homeotic genes are genes that regulate organs that develop in very, very specific parts of the body. So like for a fruit fly, for example, you say you have purple, black, green, black, yellow, black, black, blue. Well, basically focus on the colored regions, not the black ones. But like you have purple, green, yellow, blue. Purple, green, yellow, blue. Purple, green, yellow, blue. So you see how you go from the head to the tail in that order? Well, look, even for an adult mouse, what scientists realized, is that you have the same order. These same genes, because they have the ability to decode chromosomes and see what the sequences are on all organisms. They can see what the sequences are on the DNA. And they said, hey, look, we got the same genes on the fruit fly that code for like the head and the thorax and the tail. We have the same genes even in a mouse. You have the purple, green, yellow, blue in that order that will eventually be the headish, the thorax region, and then the tailish region. And then they realized that even in humans, we have that same set of homeotic genes, right? And so the way they realized that is that there's this guy, I thought this was funny, named Edward Lewis. He was the first to study this, and he found that a mutation in one of those genes actually resulted in a fly with a leg growing out of its head in place of an antenna. So what that means is the mutation was such that it wasn't that he didn't know how to make a leg. It's just that it put the leg in the wrong spot. The leg should have been the blue. It should have been yellow and then blue, but instead it was, I guess, the blue was over here with the green, and the green was maybe over there. You know what I mean? It just got switched places. But it's not like the gene for a leg got messed up or the gene for an antenna. Actually, I'm curious now, where'd the antenna go? But the point is, we have these homeotic genes that take this pattern. And so, actually, molecular studies of those genes showed that they had very similar 180 base DNA sequence, which was given a special name called homeobox genes. All right, homeobox genes, they actually code for transcription factors that activate other genes that are important in cell development and differentiation. Homeobox genes are expressed in certain regions of the body, and they determine factors like the presence of wings or legs. And so homeobox genes aren't necessarily genes that code, again, for like a leg or an arm, but they are genes that code for transcription factors. So we have to back up here. Remember, transcription factors are like the green light for RNA polymerase to transcribe DNA so that a protein can be made. And so transcription factors basically turn genes on or turn genes off. They give the green light for the gene to be read and turned into a protein. And so homeobox genes are genes that code for these proteins, these purple transcription factors. All right, so homeobox genes are very special for that. They're important in cell development and differentiation because they have that ability. In flies, there's a group of homeobox genes known as Hox genes, and that's actually what these are right here, okay? This figure 13.20 that I was referring to, these are actually Hox genes. The Hox genes are located side by side in a single cluster like this. You have the purple, green, yellow, blue. They're side by side in that order, okay? And they help determine and identify the segments of a fly's body, and they're arranged in the exact order in which they are expressed, from anterior to posterior. A mutation in one of those genes can completely change the organs that develop in specific parts of the body. So that's what happened up here in Lewis's experiment again. These got mixed. These got mixed and matched, and that's why you had a leg growing out of the head, like an antenna. And so, remarkably, those Hox genes exist in the DNA of other animals too, including us. So that's why I said it's okay to think we look like this, because we kind of did at one point early on, because we have those Hox genes too. Okay, and so the striking similarity of master control genes, that's what we call those, they're master control genes. They don't necessarily, again, code for traits, like the leg or the arm, but they code for transcription factors which turn on those genes. And so those master control genes, they have simple scientific explanation according to this book, and that is that common patterns of genetic control exist because all these genes have descended from genes of common ancestors. And so that's what evolutionists like to attribute as how evolution is similar from one organism to the next. How evolution happens from one organism to the next, based on the fact that, hey, we all have these, Hox. There's so many organisms that have these Hox genes, and so maybe it was just a slight variation in those master control genes. But I would say that the master, our master creator, didn't reinvent the wheel when he invented it, and so he had no need to reinvent the wheel and just kept designing organisms with a similar pattern. And I would say God even created organisms to do somewhat evolution while they're here on Earth, and that there is some truth to that, but not to the degree that I think the evolutionists like to credit it, or the dates and times and all that, and the sequences that they have not mapped out very well, actually. If you look at the timeline, they don't quite have that figured out. So the details can vary from one organism to other, but the switches are nearly identical. Recent studies have shown that the very same Hox genes that triggers the development of hands and feet is also the ones that are active in the fins of certain fish. Finally, there are environmental influences, and this is again where evolutionists will say and really try to help reinforce their theory that the environment also has an influence of whether or not genes are turned on or turned off. Like, for example, this tadpole, if it's in a stressful situation in such a way where, like, let's say it's in a situation where the environment gets bad, like it changes for the worse, is what it says here, then its genes will direct the production of hormones to speed the transformation of the tadpole to the adult bullfrog. So when things don't look so good, the tadpole is quickly going to become an adult bullfrog so that it can survive better. That's the idea there. Conditions in an organism's environment play a role, right? That's the point of this, what this environmental influences section is trying to accomplish. And so metamorphosis is just it's another well-studied example, again, of how organisms can modify whether or not genes are turned on or turned off, which we call gene expression. That's a fancy term for turning genes on or turn them off. And so metamorphosis, again, it involves a large series of transformations from one life stage to another. And so it's going to be regulated by a number of external and internal factors. And so that's a great way to study this. And as organisms move from larval to adult stages, their body cells differentiate to form new organs. And so at the same time, old organs are lost through cell death. So there you go. Environmental influences will have an effect. We kind of opened up with the chapter on that too, right? Talking about the lactose operon, the lac operon in Escherichia coli. E. coli, commonly known, the bacteria found in most feces. That lac operon is switched on only when lactose is present. Only when lactose is the only food source in the bacteria's environment. So yeah, environmental factors definitely play a role. And even that is not necessarily a creation evolution debate right there. That we know that to be true. In fact, that's why you'll hear a lot of people talk about why you should eat certain foods and not eat certain foods, and have particular diets, and do things like be in fresh air and sunshine and some of the pillars of health, and sleep, because those things are super beneficial for our body, because they do things like regulate genes. And so a lot of us are on